Hi everyone. Today I'd like to share with you our study titled Mesenchymal stem cell secretome in ex-vivo donor heart preservation. No disclosures. Numerous studies have reported the beneficial effect of stem cells in treating ischemic heart injury. Different cell types are under evaluation regarding their repair potential. First-generation cell types include endothelial progenitor cells, mesenchymal stem cells, and etc. Second-generation cell types mainly include cardiac origin stem and progenitor cells, as well as pluripotent stem cells. Next-generation therapies for cardiac repair are directed towards cell enhancement, such as biomaterials and cell-free concepts. Although the mechanisms underlying heart repair by stem cell therapy are not fully understood, two main hypotheses have been proposed. First is direct cardiomyogenic or vasculogenic differentiation. Second is stimulation of the reparative response through paracrine effects. Due to ease of isolation, expansion, and genetic modification, as well as safety and clinical feasibility, my work mainly focuses on mesenchymal stem cells, simply referred to MSCs. MSC-mediated cardiac repair involves release of growth and protective factors, promotion of angiogenesis, suppression of inflammation, enhancement of cell survival, and stimulation of cardiomyogenesis. Notably, all these therapeutic benefits are largely attributable to the paracrine effects of stem cell secretions in the ischemic heart. To study the rapid cardioprotective effects of MSCs, we used an ex-vivo heart perfusion system called Langendorf with acute ischemia perfusion injury that also occurs during cardiac operations, such as cardiopulmonary bypass. This Langendorf system keeps the isolated hearts alive outside of the body. MSCs were infused into the mouse heart before ischemia, following 25 minutes ischemia and 40 minutes reperfusion. You can see significantly increased recovery of LVDP in MSC-treated group compared to control. In this figure, you can see pre-ischemic infusion of human MSCs from either bone marrow or adipose tissue significantly improved the recovery of myocardial function compared to groups of the media control and differentiated renal epithelial cell infusion following IR, which was associated with reduced production of inflammatory cytokines TNF-alpha, L1 beta, and L6, and decreased apoptotic signaling caspase-3 and caspase-8 in the IR hearts. Considering the short experiment duration, a little bit longer than 1 hour, MSC-mediated regeneration and angiogenesis cannot occur, so the benefits must be attributable to paracrine effects of MSCs. Given that ischemia perfusion injury is inevitable in donor hearts due to the time window between procurement and reimplantation, we extended our study to MSC-based treatment in donor heart preservation. Heart transplantation is a key life-saving therapy for patients with end-stage heart failure, but its use is largely limited by shortage in donor organ supply, resulting in more than 1/3 of heart transplantation candidates die before suitable donor hearts are available. Therefore, there is an urgent need to develop effective strategies to increase the number of donor organs. It's knowing that short period of ischemia during safe storage generally do not cause significant damage to the heart, but the challenges arise beyond 4 hours, leading most clinical transplant centers to adapt a 4-hour time window for donor heart implantation after procurement. This limited time window renders many otherwise viable donor hearts unusable, particularly affecting potential recipients located at greater distance. We asked the question about, can we utilize MSC secretome to optimize current storage methods for prolonged donor heart preservation? Therefore, we utilized MSC-CM, that's conditioned media, and MSC-EVs, extracellular vesicles, to modify organ storage solution and investigate this modified solution in donor hearts preservation and graft outcome during extended cold storage of donor hearts. We selected 6 hours in this study. We used a mouse cervical heterotopic heart transplantation model and implanted the donor hearts into recipients. Both were from male C57 black 6 mice. The donor hearts underwent 6-hour cold storage in preservation solution containing vehicle, MSC-CM, MSC-EVs, or MSC-CM collected from MSC treated with GW4869, an exome release inhibitor. Donor heart that were implanted into recipient within 1 hour were baseline control. MSC-CM was prepared by culturing human bone marrow MSCs in serum-free basal medium for 72 hours. The collected medium was then centrifuged and concentrated 100-fold, after which it was diluted at a 1:20 ratio into preservation solution for experiment use. After implantation, we found 2 distinct QRS complex phenotypes. Red arrows indicate the donor heart's QRS, and blue arrows show QRS of native heart. The 2 QRS phenotypes were noticed in the recipients at 15 minutes, 1 month, 5 months, and even 12 months post-heart transplantation. We then detected the repeating time of donor heart immediately after operation. We observed 6-hour cold storage significantly delayed donor heart to resume their beating after implantation, while MSC-CM decreased the repeating time. Next, at 24 hours post-transplantation, we observed 6-hour cold storage significantly impaired myocardial function in donor hearts compared to baseline controls. All functional parameters in MSC-CM treated donor hearts were restored to levels compared to baseline, suggesting that MSC-CM could reverse the detrimental effects of prolonged cold ischemia on myocardial function. In addition, a higher number of apoptotic cells were observed in the 6-hour cold-stored hearts than in baseline control. Using MSC-CM reduced apoptotic cells to levels comparable to those in the baseline group. We then studied the effects of MSC-EVs on donor heart preservation. EVs preparation is shown here. First, we collected the conditioned media as described previously, and then used sequential centrifuge to obtain EVs. We confirmed the presence of exosomes in the preparation of MSC-EVs by electron microscopy. For size distribution, we found the most abundant nanoparticles at 94 nanometer as seen in the peak of histogram by nanocyte analysis. We further verified the MSC-EVs to express exome marker flattening 1 but not GM130, a Golgi marker. We found MSC-EVs preserved myocardial function against prolonged cold ischemia. In addition, using MSC-EVs reduced apoptosis in the 6-hour cold-stored hearts compared to their untreated counterparts. To further validate the protective effects of MSC-EVs on donor heart function, we utilized MSC-CM from MSCs treated with exosome release inhibitor GW4869 as shown here. We observed depletion of exosomes in MSC-CM partially neutralized their beneficial effects in donor hearts, supporting the importance of EVs in MSC-CM-mediated protection. So based on the deep microRNA sequencing analysis, microRNA 199A3P was identified as one of the most altered microRNAs in MSC-CM-treated mouse hearts following 6-hour cold storage. Therefore, we first studied microRNA 199A3P level in MSCs, MSC-EVs, and mouse hearts. We observed a dramatically higher level of microRNA 199A3P in MSC-EVs than in MSC themselves and baseline heart tissue, providing a possibility of transferring this microRNA from highly enriched MSC-EVs to the heart. Next, in mouse donor hearts following cold storage but without transplantation, we found 6-hour cold ischemia led to significantly reduced myocardial microRNA 199A3P expression, whereas MSC-CM or MSC-EVs restored this level. In addition, depletion of exosomes in MSC-CM abolished MSC-CM increased the microRNA 199A3P levels in donor heart. Similarly, in heart grafts at 24 hours post-transplantation, MSC-EVs significantly increased myocardial microRNA 199A3P levels, and inhibition of exosome release abolished MSC-CM enhanced the myocardial expression of this microRNA. Collectively, our data support that microRNA 199A3P could be transported from MSC-EVs into donor hearts. Furthermore, we detected mitochondrial structure using transmission electron microscopy in human left ventricle samples. We can see larger mitochondria with short and disrupted crista, that's mitochondrial inner membrane, in 6-hour cold stored human hearts, as shown by red arrows. However, we did not find alterations between 4-hour and 2-hour samples. Prolonged cold storage resulted in fewer but enlarged, also swelling mitochondria following 6-hour cold storage, indicating mitochondrial damage in human left ventricle. By a deeper analysis of RNA sequencing data, we found that 6-hour cold ischemia caused a class-wide disruption of mitochondria gene, particularly genes of electron transport complex 1, 3, 4, and 5. The presence of MSC-CM led to broad restoration of these changes. The gene set enrichment analysis further shows enrichment in the set of oxidative phosphorylation genes changed by 6-hour cold storage, that's left panel, but reversed by MSC-CM in the right panel. We therefore investigated myocardial levels of H2O2, a stable form of reactive oxygen species. We found that MSC-CM significantly reduced the H2O2 production in heart following 6-hour cold storage and 1-hour reperfusion, suggesting that MSC secretome likely correct the disrupted electron transport complexes, thereby improving myocardial recovery in donor hearts. Ingenuity pathway analysis of deep messenger RNA sequencing data from mouse hearts following 6-hour cold storage has identified that PER2 was one of the top differentially expressed genes, as demonstrated by the top 2 downregulated gene and the top 1 upstream regulator inhibited by 6-hour cold ischemia, shown in red bar. The results from both mouse and human hearts with ex vivo cold storage confirmed reduction of myocardial PER2 messenger RNA levels following cold storage. We also found MSC-CM restored 6-hour cold storage decreased PER2 protein levels in mouse heart associated with reduced cell death. To determine the role of myocardial PER2 in MSC-CM mediated protection following cold ischemia and in mitochondrial respiration, we used siRNA to lock down PER2 in HNIC2 cells and human AC16 cardiomyocytes cell line. The images show mitochondrial membrane potential indicated by the ratio of JC1 red to green. Our data indicated that MSC-CM preserved mitochondrial membrane potential and reduced apoptosis in HNIC2 cells subjected to 6-hour cold storage, while PER2 lockdown abolished MSC-CM-derived protection for mitochondrial membrane potential and apoptosis in HNIC2 cells. In addition, although decreased PER2 levels did not affect mitochondrial respiration function without stress, PER2 knockdown did reduce maximum and ATP-linked respiration in AC16 cells subjected to TNF-alpha or H2O2. So therefore, we summarized adding MSC secretome, including MSC-CM or MSC-EVs, to ex vivo storage solution protects donor hearts against prolonged cold ischemia and improves organ function after transplantation in the mouse model, which provide a basis to develop cell-free secretome-based strategies for optimizing current storage methods in prolonged donor heart preservation. MSC-EVs can serve as a vehicle to mediate the transfer of microRNA 199A3P to donor hearts during their ex vivo cold storage. Prolonged cold storage leads to mitochondrial injury. MSC-CM prevents mitochondrial damage from cold ischemia and restores cold ischemia disturbed electron transport complex profile in donor heart. Myocardial PER2 plays a role in mediating MSC-CM derived mitochondrial protection following cold ischemia and in preserving mitochondrial respiration in response to stress. I thank all current and former lab members, as well as our collaborators, for their contribution to this work. Thank you for your attention.